This is a special episode of the Immunology Podcast, IUIS 2023, Day 3. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rowd. Welcome back to the Immunology Podcast, where we have conversations with immunologists, sometimes in person. We're back with our third episode covering highlights from IUIS 2023 taking place in Cape Town, South Africa. Brenda and I have had so much fun attending and getting to meet all of you, but it's not over yet. If you're looking to find out what we're up to at the meeting, follow us on Twitter at @ImmunoPodcast. You can also find episodes covering the 2 previous days of the meeting at www.immunologypodcast.com or wherever you get your podcasts. Today we'll be discussing our favorite sessions throughout Day 3 of the meeting. So if you were in another session or weren't able to attend, we've got you covered. We're going to kick things off in just a moment, but before we get to that...
Are you looking to improve your T-cell therapy development? With STEMCELL Technologies' new GMP-manufactured Immunological XF, you can achieve robust T-cell expansion with high cell yield and viability without the use of serum. To learn more, visit www.stemcell.com/immunocolt-XF.
GMP. Love it, Brenda.
You gotta do it.
I'm all about GMP. All right. So, you always go to the morning keynote. What'd you see?
Oh my! Oh, what a day! First of all, what a day! So many talks, so much to see, so much to learn, so little time. But so we started strong with a keynote from Rafi Ahmed from Emory University. He is one of the fathers of T-cell exhaustion, and he did a lot of the early work in the context of chronic viral infections. And he had, basically did a really nice overview of T-cell exhaustion in the context of he started on viral infections. And he talks about the stem-like cells, CD8 cells that appear during chronic viral infection, and that they have these qualities of being pluripotent, self-renewing, that they can differentiate and kind of slowly trickle down into the effector T-cell population. And this is very important because these cells, they are the ones that kind of sustain the effector population throughout time. They are characterized by these stem-like features. They express this TCF1 transcription factor that has been is associated with a stem cell like phenotype. And they also express in tox, and importantly, they express PD-1. And these are the cells that will respond to anti-PD-1 treatment. And so he started introducing to this. He also made mention to the role of these PD-1 positive, TCF1 positive, tox positive T stem cells in the context of cancer, cool data on head and neck cancer, and the implications for immunotherapy that targeting these cells has in the context of, for example, combined with IL-2. You can both, if you combine PD-1, anti-PD-1 with IL-2, you can both remove the inhibition and kind of induce a burst of proliferation from the stem cell-like compartment, but then also sustain the proliferation of the effector cells that occur from this burst with using IL-2. So you have to go, both ways. You need to sustain them from all angles.
Interesting.
Yeah. So what were your morning sessions, Jason?
Oh, well, I don't want us to get the E on the podcast, but I went to a talk about sex.
Oh.
And sex matters. Interestingly, it was 2 male scientists who were presenting.
Okay.
But it was actually a really interesting talk. So the first presenter, Hai Ki, presented that, well known that women have stronger immune responses because you're better than us, and more autoimmunity as well, though. So double-edged sword, right? So more lupus and other things. And they really honed in on germinal cell differences between, men and women as defined by sex chromosomes, right? And so they identified that there was this one GPCR S1PR2, and it's a G protein coupled receptor that binds the inhibitory G protein GQ1213. So it's like GI is also inhibited, but it binds the G1213 and it inhibits migration. And what they found is that germinal cells will have 2 signals. So they can have a signal that says migrate towards me, right, or migrate away from me. And actually migrate away from me is how you make them in a ball in the germinal center. And so S1PR2 knockout turns off the migrate towards me signal. So knockouts have more tightly condensed central localization because they are dominated by the move away from me signal. And then they were able to look at the fact that CCL21 and this GP protein receptor 174, there's this distraction effect from the germinal centers in males. And so males are told to go wander away. And if you knock that out and also coupled to this S1PR2, if you abrogate that, then the distraction signal goes away and it drives centrally. It shows a difference in G alpha I coupling. That's the one that says go away. So more coupling in males occurs, which is more of that distraction signal. And this is B cell intrinsic. So if you take B cells from a male and put it into a female, or a female is given testosterone, it matters where the B cells come from. So if you give a female testosterone, then they behave like male B cells for those B cells. But it's about what's in the B cell conditioning, not the host.
All right.
And then this also contributes to EAE sex bias. So that's an autoimmune encephalitis we've talked about before. So GPR174 is protective against EAEs in males in a B cell intrinsic matter. It's complicated because in that model, there's a Treg component from the same receptor, and you have to abrogate that to then see the B cell, and it's an opposite effect. So you have to like unmask the Treg component to see it. The second little bit was from Zihai Li. Interestingly, also cancer is worse in men, higher incidence and mortality, regardless of where you live or your lifestyle. So I'm really enjoying that masculinity right now. It's just setting me up for future death. And they were looking at the role of CD8 T cell exhaustion in driving this. And androgens are a culprit because it transactivates, the androgen receptor transactivates TCF7, and that causes exhaustion. And loss of Y tumors respond better to immunotherapy. So then if there's all these phenotypes where, so take a step back, the Y chromosome is going away. It's just vanishing over time and shrinking over time. But if that happens, it leads to death. Even though it's shrinking, like if you lose the loss of Y fully in later in life, you have cardiac arrest and all this other crappy badness, right? But loss of Y tumors have better response to immunotherapy because they don't have T cell exhaustion as much, because that Y chromosome makes us men very tired apparently, and our T cells just need a nap.
All right.
Like I need a nap.
I see. That explains a lot. Okay. You done?
I am done with my sex talk.
Okay, good. So for my morning sessions, I went to a session on regulation of allele responses, and very interesting. Started up with Professor Megan Skies from Columbia University Medical Center, in which she was showing that in visceral transplants, in, for example, intestines, and I think also liver and other organs from the abdominal cavity. It is a hard transplant. It has a lot of rejection. But she showed that from these patients that do survive, often it happens that they observe this microchimerism in which a lot of the, more than 4% of the immune cells of the transplanted person, the recipient, are actually derived from the donor of the organ. And this actually seems to correlate better with the engraftment. So what is a long story, but what they show is that there's a situation in which you have a beneficial graft versus host reaction, in which the graft actually displaces many cells in the bone marrow, amongst them, or like immune cells, amongst them immune cells that would reject the transplant, and they establish hematopoietic stem cells from that graft. They take residence in the recipient's bone marrow, and then all of a sudden you have a chimeric person that actually can very much better tolerate the new organs that they got, and they showed a lot of data about it, but that's kind of the gist of the talk. So really cool. So kind of a good type of host versus graft.
Yeah, they should call that
reaction.
graft with host.
Sorry, it's graft versus host.
I know.
Yeah,
that graft versus host. No, this is graft with host.
Yeah, exactly. I mean, it is killing host cells, but for a good purpose, kind of. And so it doesn't cause disease. It doesn't cause a disease that kills the host. And then a second talk by Shane Gray from Garvan Institute in Australia. Also, really interesting. He talked about there's a particular enzyme called ubiquitinating enzyme called A20 that is modulates the NF-kappa B pathway, and that he showed that this is shown in there's human polymorphisms of this protein that show that in a way that's how they kind of learn what this protein does, and he showed that actually if you modulate this protein, if you overexpress it, you can use it to protect transplants, or for example pancreatic islet transplants. If you overexpress this protein, then it will protect the transplant in a way. So that's kind of cool. Of course, I'm a lot of work. I'm really making it very short here. And the third talk, I thought it was outrageous, but I really liked it. It was Bruce Blazer from the University of Minnesota, and he came and talked about his in vitro differentiated CD8 positive Tregs. Basically, this T cell he can differentiate, starting with naive CD8 T cells, throwing a bunch of IL-2, TGF beta, and rapamycin, and he can actually generate FOXP3 expressing CD8 T cells that have a considerable level of suppressive capacity. They express, interestingly, they don't express really much of CD28. They do express a lot of CD103. They express about, for what he showed in the data, 50-60% of FOXP3, CD44, and he showed that he can actually suppress T cell responses in vivo and in vitro. He expressed a lot of also CD39, and that, but this is not mediated by FOXP3 because only FOXP3 enforced expression does not do the trick. But what I thought was pretty cool is that then he made CD8 induce these Tregs like this, but CAR Tregs. So he made CARs that are both cytotoxic towards target cells, but then they are immunosuppressive towards graft versus host situation. So kind of the best of both worlds. I don't know. I'm confused. I don't know what Treg means anymore.
It's all a skid, Mark. It's just a smear, Brenda. We've been through this.
It's a smear.
You've got your ILCs, your innate lymphocytes, your T cells. It's all just a smear of biology.
It's all a spectrum.
It's whatever you need it to be. It's whatever you need to grow it up to be.
There's no truth. Oh, well, congratulations to Bruce Blazer for blowing my mind.
All right. Well, are we back to me?
Yeah.
All right. So I went to some cell trafficking.
You trafficked cells?
Yeah, the legal kind of trafficking. They may or may not have been separated from their tissue ahead of time, but that's okay in the context of cells. All right. So this was in the lab of Lawrence Zitvogel and Lisa DeRosa. The first one. So it's about how adrenergic activation seems to lead to worse cancer outcomes and intestinal atrophy, and it was ignored a long time. But interestingly, beta blockers help reduce intestinal atrophy, and that helps with cancer outcome. Well, what could be doing that? Of course, it's metabolites that alter PD-1 blockade response to therapy and just your general immune tone. And those metabolites, guess what they're caused by, Brenda? It's in the gut.
Think something funny. Candy.
It can be. That is eaten by your microbiome. And so they have a pathogenic bacteria that chews up biliary salts that makes it worse. And then if you resupplement with biliary salts, it makes it better. And what seems to be happening in their model is the standard alpha-4 beta-7 acting cells are going to go kill your cancer. There's a dysbiosis in these intestinal atrophy scenarios due to high stress, exodus of the IL-17 producers to the tumor-draining lymph nodes from the gut, and that's mediated by loss of MADCAM1. So stressed out guts, which have hyperactivation of bile acid consumption by bad acting dysbiotic bacteria, leads to loss of IL-17 producing cells in the intestine to the draining lymph nodes, and that leads to worse cancer, and you can reverse it by giving people bile salts.
Very cool.
Then I got to learn about a whole new macrophage, Brenda. The LYVE1 positive macrophages, so LYVE1. Long story short, it's a type of macrophage that seems to be involved in homeostasis and is involved in consuming collagen. So if you get rid of it, there's increased collagen deposition in your arteries, which is bad. It can be too much collagen bad, too little collagen bad, but it can deposit in your adventitia. There's lots of it in the mouse aorta, by the way, of macrophages. And it's associated with aging. So, you actually want collagen, but too much collagen is fibrosis, right? And so this is associated with aging, and it regulates your fascia collagen content. And so this is really a tour de force from the lab of Ronique Angeli, I forgot to mention the name there, on what these new macrophages do and how they seem to have if you look at their genetic program, they're not an inflammatory program, they're really a homeostatic program, and they help recycle your collagen.
Okay. So from my side, there was also, of course, the keynote from Alain Fischer, who was talking, had a very interesting, very extensive talk about inborn errors of immunity, and how to look at this and how to, I think kind of the idea of his talk was to show how the same genes can have different, completely opposite results depending on what kind of inborn error we're talking about. So he mentions things, for example, as the transcription factor Icarus, that you can have haploinsufficiency or dominant negative mutations, and that causes immunodeficiency. But there's also people that have gain of function, and then they have severe autoimmunity and defective Tregs. You can have RAG mutations that can cause SCID, but they can also cause, if it's in a very severe case, it causes SCID, so severe autoimmunity. But if you have a type of mutation that is less disruptive, then you can only have kind of partial deficiency in B cell development. So I think he was really trying to bring up the wide variety of immunodeficiencies that can occur from inborn errors and mutations that happen in few people. So in general, I think it was really good. He mentioned a lot of different diseases and how we came about to understanding the mechanisms behind them. And that was in the auditorium. I stayed over to the immune cell regulation, of course. And very cool first talk from Ramnik Xavier. He's originally from Zimbabwe, but he's working at Broad Institute now. And he was talking about, he had a couple of stories that all revolved around epithelium barrier function. So I'm going to mention maybe the first two. He showed, for example, how glycosylation and a particular transporter of manganese that affects the glycosylation patterns of mucus can have are actually a variant that is associated with a lot of diseases that are kind of inflammation related, such as, well, they see also now associated with schizophrenia, which is kind of maybe less clear, but Crohn's, for example. And he shows that, for example, the absence of these transporter results in a thinner glycocalyx that allows bacteria closer to the intestinal epithelium. This results in more inflammation, and this is kind of the what triggers the problems downstream. He also shows of a different protein, which was originally of kind of a known function, which is called TMEM258. It shows that he did some screening, and he shows that this protein, this target, is associated with epithelial stress and loss of epithelial integrity. And so after a lot of kind of studies, he pinpoints that this TMEM258 interacts or is part of the saccharyl transferase complex. And there again, the absence of it prevents proper glycosylation of proteins. And this is kind of where that's how mechanistically it affects the integrity of epithelial cells. Next up was Alexander Rudensky, who was talking about a story that we actually covered in one of the podcast roundups. So the story about this, what they call the theta cells, which are ROR-gamma T positive dendritic cells in the gut that are responsible. Apparently, they are kind of the missing link in the early induction of ROR-gamma T positive Tregs in the periphery in the gut. And this is a story from Chrysanthemum Brown as well. And so I remember, so we discussed this, so I kind of sort of familiar with the story. But basically, they really try to pinpoint where are these Tregs getting their signals from, and why is it so important. This is really early on kind of the post, very early age after birth, and they really pinpoint down these ROR-gamma T positive dendritic cells. And then he had another story about Tregs and how they interact with nociceptive neurons and how they modulate the pain receptor and sensation. And there's the expression of a precursor of an endogenous opioid neuropeptide called PENK, and that this Tregs in skin express PENK, and this PENK protects the pain neurons from overstimulation and therefore have kind of analgesic functions. So elimination of Tregs in this context increases pain and discomfort in the mice. And last but not least, Christophe Benoit, also very well known in the Treg field. He had a lot of stories. On the one hand, he was talking about how there's a certain inheritance of if you mix BALB/c and BLIC6 mice, they're already known for having different proportions of ROR-gamma T positive Tregs in the periphery, and this is kind of strain dependent. But apparently, what he does when he tried kind of combining the strains, and he actually showed that it's not so much strain dependent as it's mother dependent, and he shows a kind of a direct relationship between the levels of IgA in the gut of the mother with the development of Tregs in the pups. And so there's always the mother, regardless of the genotype of the mother and the father, the pups kind of inherit the Treg proportions of the mother, and this depends on this IgA repertoire that they already have. And talks about also specificity of Tregs in the gut, again, trying to understand what are they recognizing. He shows that Tregs in the gut can recognize bacterial antigens, and that there's something about the TCR itself and the sequence of the TCR that will kind of promote the differentiation in a Treg phenotype, which is kind of weird. So they put Treg TCRs on conventional cells, and apparently those are Treg-derived TCRs are better at inducing Tregs on conventional cells than non-Treg TCRs. So I thought that's also kind of cool. Just want to say one more thing. There was a lot of neurons and immune cell interactions. There was this aging and neurodegenerative disease, Vera Gorvunova from the University of Rochester, with a story about transposons in aging and how as we age and we have more DNA damage, our sirtuin 6, which is a histone deacetylase, that during our youth is busy keeping a particular transposon called L1 in check and preventing expression. But then as we get older and then sirtuin 6 is busy with other things, then we start expressing these transposons, and the accumulation of transposon cDNA in the cytoplasm activates SIGA-STING and generates inflammation. And this is part of like kind of the inflammaging phenomena and these weird transposons that are making us old. Really weird. So yeah.
There we go.
Packed sessions.
Packed. Well, that brings us to the end of our latest IUIS 2023 episode. Don't forget to follow us on Twitter at @immunopodcast to find out what we're up to at the meeting. Check back here tomorrow for another episode recapping day 4 of the meeting. See you then.